Welcome to today's Infection and Immunity BSc taster session. Thank you very much for joining us. My name is Andy Wei. I recently completed my Infection and Immunity BSc and I'm your chair for the event. We hope this taster session will address any course-specific questions you may have and help you gain an insight into what it's really like to study on our Infection and Immunity undergrad program. So this afternoon, our speaker, Dr. Douglas Fink, will introduce you to some of the great science and medicine that you would learn as a student of the Infection and Immunity BSc here at UCL. Dr. Fink will also provide a summary of the Infection and Immunity program, and there will be a Q&A session for the second part of the event. This session is being recorded and will be made available on our website following today's event. We're here to respond to your questions, so please share those throughout using the Q&A function on Zoom. Now to introduce our speaker, Dr. Douglas Fink is a clinician scientist working between UCL and Royal Free London NHS Foundation Trust. He also works with the Nigerian Institute of Medical Research in Lagos, Nigeria. Over to you, Dr. Fink. Thank you.
Thanks, Andy, and good evening, everybody. Just to say, if you've got questions, as Andy highlighted, please do put them in the chat, and then we'll come to those when we come to the Q&A. And it's great to see so many of you. So thanks, Andy, for the kind introduction. This is going to be pretty informal, but at least some of it will give you a flavour of the type of material we'll cover. My background, as Andy said, is mostly in infectious diseases and the immunology of infectious diseases. So we won't go into a lot of detail around non-infectious immunology today, and maybe we can touch on that at the end. But the division, as the name suggests, doesn't just focus on infection. We're interested generally in the broader aspects of immunology, which encompasses some cancer, autoimmune disease, and as the title of the division says, transplantation. UCL, as you may be aware already, obviously, because you've got as far as this taste session, is right in the middle of town, and we can talk about the location and the other sort of environment factors that might persuade you to come learn with us. But it's a really great campus. What I'll run through, as I said, is some science, so you get a real sense of what you'd be learning with us. I'll also go through the content of what our degree looks like. This says you'll hear from two students, but really that's just Andy. But he's like two students. He's that effective. And then at the end, as I said, we'll have plenty of time for question and answers. So as we go along, store up your questions, put them in the chat, and then we can address them at the end. So as I said, my interests generally are in infectious disease, but more specifically in viruses. And I think most of you will be familiar with the headlines. This last week, obviously, we've had hantavirus in the news, but obviously the last few years have seen many different high-profile infectious diseases, no less than COVID. But what's a remarkable thing about these pathogens is that really they're just a clever sequence of genetic material. And stripped down to their bare bones, there are a piece of genetic code in a container shown by this cartoon. But what they do unbelievably effectively is hijack human cells, or indeed other cells from other animals, to turn humans into factories for their genome. So we become virus factories where we're infected by viruses. And you can really understand a lot about how humans work by following viruses. And we also often talk about viruses being the cleverest entity in the room. But on their own, they don't really do anything. So fundamentally, it's the interaction between viruses and their host that causes disease. So this is a simplified life cycle of HIV, which is until SARS-CoV-2, was by far and away the most studied virus. Caused pandemic disease, affects something like 40 million people globally. And what you can see, like all viruses are required to do, they have to be able to enter cells. They have to then be able to replicate their genome. And in the case of HIV, that requires a magical enzyme called reverse transcriptase that converts RNA into DNA. The amazing thing about HIV in particular is that it literally becomes part of the people it infects. So you can see here that the DNA in red at the bottom becomes part of what's called a provirus by inserting itself into the human genome. So when you're infected with HIV, your virus literally becomes part of your own DNA, which is quite remarkable. And with only 9 genes, it's able to turn again the cells into factories for virus, produce all the proteins it needs to do to build new particles, and it's that goes on to infect the next host. The reason we show this is really this paradigm of cell entry, genome replication, gene expression, and particle synthesis is really the core of any life cycle of a virus. And to be successful, that's any pathogen, particularly viruses, have to be able to do that. But what's clever is this essentially arms race with the cell, because we, as we've encountered these pathogens, these viruses over time, have evolved mechanisms to detect and then destroy, or at least interrupt, the life cycle. And one of the key things that affects this, and you can see it's symbolized in this cartoon by these yellow triangles, is something called interferon. It's a pretty cool name for a host response, but interferon gets its name from flu, where we found that in the sort of middle part of last century, that actually there's something in the supernatant, in the fluid around cells when they're infected by flu, that if you place it on another set of cells, stops those cells from becoming infected. So it interferes with infection, thus interferon. So it's really the first host response to infectious disease, and it's this incredibly clever mechanism that I'm going to show you in a second. So HIV, as shown is here comes into the host cell, but your host is prepared and starts becoming these interferon proteins that then protect it from infection. What's even cleverer is it doesn't just protect the cell that's infected, but it essentially sends up an alarm signal through the protein that then can alert neighboring cells to also upregulate their antiviral responses. And so this establishes something called the antiviral state. And it's really effective when it works, and it blocks viruses from infecting neighboring cells. But then beyond that, the arms race continues, and actually viruses evolve to counter these mechanisms by also producing things like proteins or other products that prevent the interferon from working. So we call that immune evasion or immune suppression. So in any evolution between a human and its pathogen, you see over years, maybe millennia, but even over short years in the case of SARS-CoV-2, this arms race evolutionary battle between the virus and the host, one evolving to evade the immune response and then one developing immune responses to trap the virus. So HIV, as I said, was the sort of paradigmatic virus that we used to work on a lot, and that's kind of a lot of my expertise. But obviously 5 years ago, a little over 5 years ago, we had the more recent pandemic with SARS-CoV-2. And these are my old colleagues, Lucy and Lorena, working in a Cat3 facility. And again, I just wanted to show you the sort of work that we actually do at UCL and the sort of science that you'd be exposed to. So again, how do you even measure viruses? Well, one way you can do it is by labeling them. So you take a virus and you genetically modify it such that it expresses something called a green fluorescent protein, or GFP. And then what you can do is just look under a microscope. So at the top here, these are some cells that have been taken out of a human's airways. And at the beginning, you can see there's no obvious viral replication. But by the time you get to 10 hours, these cells that have been exposed to SARS-CoV-2 start expressing GFP. And that's because the virus has got in, it's expressing its genome like we saw for the life cycle of HIV. And because we've changed its genome, it now expresses the green protein that you can measure through microscopy. And then that's what it looks like after 24 hours. So a lot of the research we do is finding clever ways to measure viruses, because ultimately you can't test whether a treatment works or whether a vaccine works unless you can measure the virus. So we're really interested in that. Yes, I mean, this really is just to highlight that actually viruses are definitely the smartest entity in the room, and they're the thing that outstrips even the best scientists. So during the COVID pandemic, the virus was continually evolving around not just our response without vaccination, but actually also to the vaccination that we provided. And so, using those mechanisms I've just shown you to measure virus infection, what was interesting to Lucy and Lorena was that the original coronavirus um, was pretty good at replicating, but you can see actually, if you think of the amount of virus on the y-axis here, so high levels mean that there's lots of replication. So it doesn't reach its peak of replication until 3 days, so only 2 hours after you put you put the virus on the cells. What was interesting about the second virus that emerged, the one that evolved, is that actually it was able to replicate much faster. So you can see here, if I use my pointer, hopefully you can see it. In the sort of browny reddy color, the peak of the virus was actually within 48 hours. And what that means actually is that the virus can transmit much more quickly. So viruses transmit to other people when the viral load is very, very high, because that allows high levels to be emitted into secretions like when you cough or when you sneeze. How did it do that? Why was this virus different? Well, actually, it turned out because it was different in its ability to interfere with interferon. So, what was found out was that actually, if you look here on the right, the bars are for two different doses of the original coronavirus. And again, in this graph, we're looking at the amount of interferon produced measured through an RNA measurement. So this is basically saying how much interferon is being released by those cells exposed to virus. Whereas remarkably, even incredibly high doses of the new virus actually didn't induce hardly any interferon at all. So what that means is the virus has adapted to the human host and has enabled it to get in and out, producing very high levels of virus that you can see on the left, but without inducing any kind of immune response. And that's incredibly effective as an evasion strategy to increase transmission. So this is what the paper actually looks like, but as you'll probably be aware, Nature journal it is the most high impact science journal. And just to give you a clear sense, we've taken a concept, applied it to an important clinical problem, and produced really high quality science that fundamentally has changed how we understand viral evolution. And that's all happened in our division. You can see that picture I showed you a second ago is literally in this Nature paper. Obviously, the story doesn't end there. It turns out, actually, as all of the viruses evolved, if you look first on the left, this is just a cartoon which reports on the y-axis the percentage of the virus sampled in the population at one time, and then obviously the dates, the time of the pandemic on the x-axis. So the different colors of the different mutant coronaviruses. And so it changes over time. It's a very dynamic process, but it's very clear that all of the descendants of the original virus, which here is called VIC or IC19 in the bar chart, all of them produce much lower levels of these immune responses, which are measured in this graph by something called CXCL10, which is a protein your immune system responses. Your immune system produces in response to virus infection. And this was produced by another colleague, another friend called A.K. Royschel. I really should have put a hantavirus slide on here, shouldn't I, Andy? But again, this is just to illustrate all of the sort of dynamic infectious diseases that we have to deal with in the hospital where I work, but also trying to put these into the context of immunopathogenesis, trying to come up with new treatments, new tests, and new vaccinations. So Mpox was something I looked after a lot of cases here, and then we're just beginning to understand the biology of. We looked after Ebola cases here as well, and again, there's now an effective vaccine because of the effective research, and we continue to worry about the next pandemic and influenza transmitted from birds. So-called avian influenza is definitely a commonly cited example of a potential pandemic pathogen that we worry about. And then I guess I just wanted to highlight where the future holds. So the world is getting smaller. I think you all know this already, but obviously travel means that we can get anywhere in the world in a matter of hours. So pathogens, viruses that used to hide many many days or weeks or months travel away now can travel across the world in hours. But further than that, because of the way cities are organized, how urbanization works, we're now expanding our civilizations into previously uninhabited spaces. So these jungle spaces, which have lots of wild mammals and other animals such as bats, are now being disrupted such that the pathogens these environments carry are now exposed to humans. So it's very clear that the COVID pandemic was caused by deforestation in China that led to exposure of bats and their coronaviruses to humans and evolution of SARS-CoV-2 that spread around the world. And what we're trying to do using some of the information from Lucy and Lorena and other people is to try and work out how we can use AI and other technologies to predict what the next pandemic might be, and also potentially, in theory, generate vaccines even before these viruses emerge. But the ideal scenario is that we start going into these wild parts of the world, sampling, looking for things that might cause trouble, and then developing treatments in advance. Okay, so that's a kind of flavor, obviously more virology, immunology flavor. I think the key point, and we can come to this when we discuss at the end, is that our kind of philosophy, our principle in the department is to treat you like adults and try and expose you as early as possible to the actual core science that makes our division successful. So rather than us teaching you from textbooks, we're trying to expose you directly to the sort of laboratory work that's actually being conducted in real time. So when you come and study with us, really you're at that frontier of where these different disciplines are. And that's pretty unusual in an undergraduate course. We can talk about what the benefits of that are. But my feeling is it really makes you feel and understand science as a profession. It's a way of discovering things in a very structured way, and that can feed directly into things that have a big impact on the world. So let's just look at the course in a bit more detail. Again, plenty of time to discuss the details of this when we come to the Q&A. But again, this just gets across what I think is the core principle, is that we really want you to see science as a career, not just as something you need to learn. And to understand that the process of discovery science, discovering new things, is a creative process. Yes, obviously it needs to be structured, but actually the scientific process is all about testing hypothesis, generating and then testing hypothesis. Yeah. How does the actual course support this? At the beginning, most of the first year is taught where I am at the moment, which is in North London, in Hampstead. I promise you it's much more attractive than this picture implies. The Royal Free Hospital may be the least attractive structure in Europe. But actually where we do the teaching is quite nice. And we've got a brand new immunology building up here called the Pears Building, which I should add to these slides. But the point of the first year really is to give you an enhanced understanding of human biology that goes beyond what you've learned at A level or baccalaureate or the equivalent. So you cover a whole load of core modules that really gives you the foundation to understand how infection and immunology interfaces with these things. I mean, our argument is that the whole body is governed by the immune system. And what's becoming clear is almost all disease is governed by the immune system. So almost all disease now has a treatment that is based on what we call immunotherapy. So harnessing the immune system or repurposing the immune system. The first year is pretty broad. It will feel related to it won't feel like a huge jump from your school, but it really gives you a foundation to then go on to the more complex science. Year two, again, this was where it becomes a little bit more laboratory focused and more, I guess, molecular basic science. So again, the key point is we're trying to develop you as professionals. That means exposing you to actual scientists, teaching on their actual subject that they're literally working on in real time, but also developing those hard skills that are transferable whatever you do. And critical analysis and communication are core to that. So the ability, as I said, to generate a hypothesis, test it, then present that data, which we call dissemination in science, but then also to analyze it. And there's many ways to do that. And those are principles that you can take really to any work that you do, and we'll look at that in a second. So the idea is you're no longer just receiving information. You're not a school kid anymore. You're there to begin to process it and bring your own understanding and experience and interest. We also have a formal journal club. There's also the opportunity to do literally hands-on laboratory work. So it really is much more focused while also giving you these broad skills. And you'll also begin to have the opportunity to do some more optional modules. Year 3 is where it gets most exciting. I think Andy probably agree with that, but you can correct me. There's still some core elements, but it's much more specialized. And then you have lots of different options. And a lot of these are quite translational. So you might learn about how we develop drugs to treat autoimmune disease, cancer, something like that. In modules that I run, we focus more on viruses and generally infectious diseases, but we also potentially connect you to working in again, the translational space, generating interventions for those. And the key point is you do a research project. So really your whole, not quite, but really at least half your year really is focused on literally being either in the lab if you want to do laboratory work, or working with a computational team if you want to do in silico computer-based work, and really taking ownership of a project. And generally we can pretty closely match your interests to people in the division and even outside potentially. Move that on. I think it's important to think, although must feel like a long way away, to think about where this degree takes you. And yes, you'll end up with some pretty specific knowledge and expertise in infection and immunology in the context of both health and homeostasis, but also disease. But you also really broad scientific training, and really analogous to almost any undergraduate training. That could be biomedical science or one of the other undergraduates that you might also be courses that you might be looking at. So I think it still gives you those core skills that you can apply to whatever you decide to do afterwards. I think what we're particularly good at is really teaching you those hard skills of critical analysis, data interpretation, presentation, communication, and then potentially actual real lab work. And we base a lot of our assessments on actually testing those. So you'll be used to doing written exams, but actually we really like to do exams that work on your ability to present a paper, present a project. And although that can be pretty, daunting at the beginning, I promise you that's about the most useful thing that you can do. And it's so valuable to whatever you end up doing. Also, obviously, you'll write a project. And so there's the opportunity to develop your writing as well, which is obviously transferable. Here's a bunch of people who've been through the program either as undergraduates or intercalated from medicine. So James Kai there in the middle on the right, he was a student of mine who is now a doctor, but he came as an intercalated student. So it's worth just mentioning that what's nice in the third year is that you also work very closely with the medics. And I think it's really nice to get a slightly bigger year group and a bit of breadth of experience and personality. So it ends up being something like 15 to 20 undergraduates with about 10 to 15 intercalated BSc students. But yeah, what's happened to people? All sorts of things. Lyell at the bottom left there, she's gone on to be a really successful science writer. She published a book 2 years ago. Many people go into master's programs or even PhD programs. Andy can speak to that. He's currently in that zone at the moment. And then all of those sort of science or medicine allied careers, so pharma, consulting, and people have gone on to things that don't obviously feel related to science, so law, because the skills you acquire really are so transferable. So you shouldn't think of this as a narrow course. That would be a mistake, I think. If you want it to be narrow and lead to a specific track, it will, but it really gives you a real breadth of training across science and those other transferable skills. Yes, so the prospectus, no doubt you've seen already. that is the standard offer. Biology and chemistry, as I understand, are still required. Um, Mathematics is definitely not essential, but it might make your life easier. And then obviously, again, there'll be physical open days, which you'd be welcome to visit, and you can book a campus tour. And it's a great campus. I mean, we'll talk about that, I'm sure, in the discussion. But you know, really, it's in the coolest part of London, pretty much. You can walk into Soho in 10 minutes, but you can equally get on the train and be out of London in no time at all as well. It's really well connected, and the immediate environment's nice. Uh, cool. So, um, again, I just wanted to go into this, and then I'm going to go back to that previous slide, and we'll start let Andy um, do some talking. Um, so yeah, as I said, really, it would be misleading to think of this as too narrow a degree course. Um, it's really similar to the other biosciences programs, and so you could apply it to lots of different careers. The transferable skill set, I think, we do to a really, really high level, and I think that's consistent feedback. Um, if you really are passionate about infection or passionate about immunology, then there's, I think, certainly no better undergraduate course for you. But it doesn't mean that it closes off other avenues. And that core thing is that you're going to get taught by actual scientists doing actual cutting edge research, and that's pretty unusual at undergraduate level. So I think that's all I had to say for now. So I'll stop sharing actually, and hand over to Andy. So I think Andy, probably you've got something to say, but I'd be really interested to hear. You just got to give a little bit of feedback on your own experiences. Thanks.
Thank you very much, Dr. Fink. So for me, this course was really well thought out. So from year one, you get to learn about the entire body, how different organs work, how they work all together. And then in year two, you get a bit more specialized in infection and immunity. But you also get that optional module where you get to choose from different other topics like evolution, mathematics, modeling, and there was also one in, oh yeah, clinical trials. And in year three, you basically focus entirely on infection and immunity for the core modules, and you get optional modules in cancer, autoimmunity, mathematics modeling again, and evolution, viruses. So it's really tailored to your interest. And also the big highlight was the research project in year three. So essentially you get to work on a research project that is tailored to your interest too. So if you're interested in cancer, you could work on something related to cancer. If you're interested in viruses, you get to work on something related to viruses. And the most important thing is that you can learn from the people who are doing the research, the state of the art research. And that's something that you won't get elsewhere. You rarely get elsewhere. So now we have a couple of questions for you, Dr. Fink.
Thanks, Andy. I can see. Shall I do the? I can see the first couple
Oh,
ones, which are
great.
actually quite
Yes.
pretty sophisticated questions, pretty interesting questions. And there may not be a super simple answer, but yeah, great question. So Pooja's asked, what role does capsid play in viruses? So that's a really good question. So capsid is generally thought of in most viruses as the inner core. So generally, you have what's called an envelope for some viruses, and then within the envelope, you then have a capsid, which is multiple proteins that are called capsid that form this sort of canonical structure that basically is a carriage mechanism, a container for the genome. But it turns out actually those proteins have lots of other functions. So in HIV, which is probably the best studied capsid is not just a container, but it also interacts with all of these host factors that allow the genome of the virus not just to travel protected, but actually to start shutting down bits of the immune system. that might otherwise attack it and stop it replicating. So, yes, in HIV, there is a drug called lenacapavir, which is actually licensed for treatment. So it targets capsid by stopping it from forming and potentially causing it to fall apart. But actually, there aren't that many capsid-directed agents in other virus infections. So, Pooja, that could be your area of expertise eventually. Um, and then someone's also asked, what signs do you look for in possible pandemic pathogens? What's most quickly how it mutate, or are there other things? Yeah, so really complicated question and an interesting question. And there's two things, I guess. One, it's about looking in the environment and finding pathogens that have the potential in the future to cause pandemic disease. And almost all pandemic pathogens are zoonotic. So they start in an animal and then they jump into a human. And the reason they become pandemic is because the human race is completely naive, i.e., has no immune memory or immune response to those pathogens. So the virus essentially can run riot, produce very high levels of viral loads, and then that means transmission is very effective. So HIV was zoonotic, Mpox zoonotic, SARS-CoV-2, SARS-CoV-1 obviously zoonotic. So that is an important thing. So if we're screening nature and animals looking for those pathogens, that's an interesting approach. But then once something gets into humans, what makes it more likely to be pandemic? I think, yeah, absolutely. So coronavirus is a good example of something that gets in and out of the cell super quickly because it's an RNA virus and it replicates not in the nucleus like HIV, but in the cytoplasm. So it can turn itself around really quickly. Whereas HIV is a good example of something that mutates. So in any one person with untreated HIV, there are more HIV virions than in the entire population of planet Earth in terms of humans. And at any one day, it introduces about 1 million mutations into that population. And that means it's very difficult for the immune system to catch up with it, which is why HIV is so effective as a pathogen before treatment, and why there's no vaccine, and why ultimately you don't become immune from infection. Um, so I'm not sure that answered the questions, but yeah, I think when we're looking for new pandemics, we need to get out into nature. And when we're looking at mutation, yeah, there's a few different flavors to that. Right. Okay. Next one's, would infection immunity come under biomedical sciences? Technically, it's a different degree, but it is basically very similar in terms of the first year, at least. And it would basically should lead to a similar journey. Okay, this one's for you, Andy. I think I'll hand over to you for a couple now. So Takaya, I think, has put a question there.
So we're looking, the question is, what are supercurricular activities that students tend to take on who are successful in their applications? So when I applied to the course, I did some extra reading. So on papers, uh, that you might be on topics that you might be interested in, although I know it's quite difficult at that stage, but also just reading around any infectious diseases or immunology stuff around the internet. And then there's also courses on the internet that you can complete offered by different universities, or it doesn't even have to be a university. Um, so you can look in some cancer courses, autoimmunity courses, or infectious diseases courses. So these are quite helpful.
Thanks, Andy. Yeah, I think that's right. I mean, when it comes to writing your personal statement or your application, really, you're just trying to tell a story that emphasizes why you're the right person for the course and why you're so interested. And good evidence of showing interest in you're the right person for the course is showing that um, you've read around, as Andy says, and you've kind of demonstrated that you're really interested in science. So don't worry too much about, I don't know, being a international tennis player or something. I think we're more interested generally in people who, on some level, have been able to tell a story. They're interested in science. But yeah, why is maths recommended in the course? Is it necessary to do well? So, I mean, Andy might be able to contribute to this. I'd say it's definitely. helpful, but actually, if you're doing chemistry and you can cope with the maths and chemistry, I think that's more than adequate for you to do this course. And what you'll find actually is when you start apply, if you don't enjoy maths, what you'll find is that when it moves from being the theoretical maths of school, when you start actually applying it to your own experiments or actual data and thinking how you might use it, how you might make use of it, it actually becomes pretty interesting. Um, so yeah, Andy, I don't know what you think about that, whether you'd agree.
I completely agree. I think um, the professors here really, really will help you like take from the very beginning for maths courses and modules that we learn. So any stats we learn from the very beginning. So really, although it's recommended because it will help you understand maybe better, but really everyone's here to help, even if you don't take maths. And because the support that's here, you don't really need it to do well, although it will help with your studies. And I
Great.
do completely agree that when you actually use the maths in your experiments or interpreting results, it gets much more fun than all that theory that you learn.
Great. Um, Next question is, could I apply for the NHS scientist training program with this degree, and how similar is this degree to biomedical? So yeah, I mean, I'm not sure we've got a specific example of someone who's gone through onto that program. I think it's pretty new, but 100%. I mean, it's a very, very similar skill set. And I would argue that you'll probably end up with better hands-on science training, which is directly applicable to that NHS scientist training program. Um, so yeah, I wouldn't hesitate. Um, Sakai has asked again a bit of SARS-CoV-2 biology. So, what is it that the alpha variant of COVID virus did to suppress the release of interferons? Yeah, good question. So, essentially, without making it too complicated, as I said, there's this state of co-evolution, this arms race between your virus and your humans, who are continually changing the way they interact. So what viruses tend to do is either they evade interferon, and actually the capsid of HIV is a good example of that, in a way that the capsid protects the viral genome from being detected. So if you don't make any interferon, so if you don't get any detection, then the body doesn't know that it needs to make interferon. But in COVID, what it does is actually produces other proteins that target different bits of the interferon signaling pathway. So there's lots of different um, proteins within humans that contribute to the signaling that results in interferon. And viruses are super clever and they know exactly which bits of that pathway to target. So, yeah, what that virus did was it started making lots of a particular type of protein. And it basically just obliterated the interferon pathway. So, if you think of like interferon as our radar defense missile system, and the virus starts working out how to target key parts of it that brings down the iron dome of interferon. Yeah. All right. Maya's asked, what made you choose this study? What made you choose to study this degree over other similar ones? If the content's similar, that's to you, Andy, I think. So why I&I over biomed, essentially?
So for me, I liked because I&I is within medical sciences, and medical sciences is more focused on the human body, whereas biomedical sciences you have modules in chemistry, so you know organic chemistry and other biology modules that could be less focused on human physiology. And another aspect was the research project that you get to work on something that you're genuinely interested in, because it's a relatively small cohort, and you get to work with the professors that are doing the actual science. And I think that's really something quite special about this program.
Thanks, Andy. Yeah, I think that's the key. That's a really key point. Um, Alex has asked, what first brought you to Nigeria, and how does its infectious disease compared with other places you studied or worked in? Yeah, so yeah, clinically, I manage people with infectious disease, and I've worked in East and West Africa at points. The original interaction in Nigeria is with their medical research institute trying to understand um, basically non-infectious disease in people living with HIV. So it turns out if you have untreated HIV, your immune system gets compromised, and actually that leads to all sorts of non-infectious problems, which can affect your lungs and infect your heart and infect even your bones and joints. So that was the original reason, and then we've done lots of projects over the years, including in COVID. Um, and, obviously, we're lucky in the UK, we have a very well established national healthcare system and surveillance system. And actually, Nigeria has a very good now national surveillance system, but ultimately, there's much less resources, just less money support that. And obviously, in that climate and in that environment, there are different viruses, including viral hemorrhagic fevers, things like Lassa fever, for example, that we don't have in the UK, although we occasionally have to manage them when we come here. So, and malaria is obviously another classic example of a infection associated with mosquitoes in the tropics, because although we've got mosquitoes in the UK that have the capacity to transmit malaria, in fact, the last case of malaria transmitted within the UK was in the 1950s, and it turns out malaria used to be called marsh fever in the UK. But the reason we don't have malaria transmission here is because of the climate. And the fear is that as the climate changes, those mosquitoes, literally because of the temperature, will then be able to sustain the malaria life cycle, and we'll end up with malaria again. So a nice interaction there between what we often call One Health, or global health, and climate health and infectious disease. Iman's asked, how does HIV research contribute to cancer immunotherapy? Yeah, that's a pretty complicated question. So as you guys probably know from your A levels or equivalent, HIV targets these key immune cells in your body called CD4 T cells. It literally needs the CD4, it's a receptor, it's basically something that sits on top of those cells, and it needs that as a way to get into the cell. And over time, what it does is it kills those cells, and you die from HIV because the virus wipes out your immune system. So very quickly, back in the 1980s and the 1990s, the world had to really grapple with the immunology of T cells, and so basically use the virus as a way to understand T cells. And a lot of that knowledge around T cells has now led to the ability to manipulate them to treat cancer, either by drugs that target the cells directly, or actually by taking the cells out of the human body and using gene therapy. Beyond that, gene therapy literally takes viruses that are related to HIV or even derived from HIV to deliver bits of genetic code into cells. So gene therapy often actually means an HIV delivery system. So our knowledge of immunology, and then also literally the tools we use to manipulate immune cells, have derived from HIV research. Yeah. To what extent do researchers currently think chronic low-grade inflammation contributes to systemic diseases rather than being simply associated with them? The example is bacterial pathogen and Alzheimer's, essentially. Yeah, it's an interesting question. Lots of active research in cognitive impairment and dementia, really important problem that we face in an aging population. And not my particular area of expertise, but certainly something that people are thinking about. Because again, things that cause long-term inflammation leads to dysregulation of your immune system, and then that can start targeting your body and including your brain in a way that's unhelpful, it's harmful. So yeah, not something that I know much about, but certainly something that's actively researched. We often hear about bacteria becoming resistant. This is from Nolan. Can viruses also develop resistance, for example, to antivirals? Is this probable, quite rare, talking in the future? Good question. And no, actually, it's pretty common. The key point with HIV is, because it evolves so quickly, is that in the first trials, for example, we used to use a single antiviral agent, but the virus within weeks or months evolves around it. So what we now call antiretroviral therapy actually usually is a cocktail of at least 3 drugs. So rather than just putting the virus in handcuffs, in a sense, you're handcuffing the virus, chaining it to a wall, and putting a hood over its head. So you're targeting lots of different parts of the life cycle, so that even if it manages to become resistant to one part of the drug regimen, it won't be resistant to all of it. But no, it is a genuine problem in viruses as well. But we've learned that using multiple different antivirals at the same time in certain contexts is the way around that. Okay, back to the course. So better ask those ones. Parts of the course you find favourable, and what led you to this course? I think we've sort of answered that one already a bit. Um, this is comparing to biomedical sciences, and they're unsure. This is anonymous, but unsure if infection immunity course is the right one for me. Is it possible to start with biomed and then specialise in infection immunity? Um, so at the moment, that's not super straightforward. I hope that we've reassured you that actually starting in infection immunity is the way to go. And really, in terms of your outcome at the end of your degree, it can be really the same, and I wouldn't get too anxious about that. If you're interested and you want to do hands-on science, I think I&I is the way to go. Um, Andy, is there a lot of group work or is it mostly individual work? Do you want to have a go answering that one?
I think there is a balance. So you do get group projects, at least for the first year and second year. And then in third year, I don't think at least when I did it, there is mostly just individual work. But there is a good balance. So you get to, make new friends, interact with different people, hear different opinions, and that can be very interesting and very helpful.
Right. Takai has asked, how should I phrase my personal statement if I'm applying for this course, um, but for biomedical sciences at other universities? I'm not sure there's a I wouldn't worry too much, really. I think you just need to add a couple of things that maybe give us a sense you're particularly interested in infection or immunology. But ultimately, it's going to be pretty similar. And actually, being interested in science is probably more important at this stage. Andy, do you remember how you balanced that? Because presumably you also applied to other places with a slightly different um, course emphasis.
Yeah, so I think I had a emphasis on infectious diseases and immunology because that was what I was genuinely interested in. And I think it also helps with biomedical sciences as well because it's part of the topic. And it's really just showing your interest and I would say love for science,
I
which
think that's
really
a good point.
attract.
I think actually, if you're just showing interested in science, and that might be infection immunology, then that'll absolutely be relevant to biomed at other places, because infection and immunology are a big part of those degrees anyway, as Andy says. Yeah. Okay. Amy says, how did you realize you were interested in infection immunity specifically? Were you confident you wanted to specialize in it from the beginning? Andy, do you want to answer that one?
So that was from my secondary school biology experience. So during A-level biology, we did some experiments in infectious diseases and also immune cells. And so that was really, really what interested me to take on this course from the beginning. And also, I think COVID. So when I started, it was during the pandemic, if I remember correctly. And that really kind of sparked my interest.
Yeah. When you get started, yeah, you'll be even more convinced. But yeah, immunology, to be clear, is not always infection. So if you're interested in cancer, autoimmunity, transplantation, and as I said, almost anything, even we touched on dementia, for example, immunology is core to all of that. Um, what modules and tasks do you do if intercalating into this degree? So if you end up doing medicine and you intercalate in infection immunity, it's exactly the same as it is for the undergraduates. So we make sure you do a project. There's a handful of core modules in the third year, and then lots of optional modules. So it's pretty flexible actually in the third year. Um, so it's Sally. Are there supercurricular activities you recommend for Year 12 students interested in immunology? Andy, you kind of touched on this already. Do you want to say again what you think is useful? Maybe online resources that you'd recommend?
Yeah. So I guess just any reading that you can do around the topics that you're interested. So say if you're interested in cancer, you could read around some papers, some textbooks, or even on the news about cancer, like new cancer drugs being developed. Um, so it's just really to show that you're interested in the topic. And I
Yeah.
think that's was crucial.
Question about infections and temporary kidney injury, and then chronic kidney disease. So again, uh, almost too much breadth there to focus on one answer, but obviously the real answer is that often it's the infection that causes an initial issue, but then actually it's the immune response that leads to the disease. So although the immune system might be attacking the virus, the bacteria, actually an overexcited immune system can also damage your organs, and that includes kidneys potentially. So it's a very simplified answer, but yes, often it's actually your immunology that leads to the kidney disease in the context of infection. What's your opinion on the hantavirus outbreak? Is this the kind of thing research in infection immunity? Yes. So this morning I had a meeting around trying to get samples to develop new monoclonal treatments, antibody treatments for hantavirus, because there aren't any at the moment. And then once I finish this call, I will be finishing writing a treatment guideline for the outbreak. So it's a virus that we traditionally think of as a zoonosis, as we touched on. So it comes from rodents and into humans. And what was unusual in this outbreak, although it has been seen before in Chile and Argentina, is that it was spreading between humans, or at least we think it was. Um, but I think the reality is that probably it was a perfect storm, and the fact that they were in such close environment on the boat. So I think it's pretty unlikely at this stage it's going to go beyond the population on the boat. But we're always surprised. But absolutely, it's exactly the kind of thing that we're interested in infection immunity. Could overuse of antiviral drugs lead to antiviral resistance? Yeah. So, and the difference with viruses tends to be that they're not environmental generally. They're not commensal. Bacteria, if you introduce resistance, then they get reintroduced back into the environment. They essentially can circulate and exist, whereas viruses don't tend to do that in the same way. So generally, resistance problems with viruses happen in an individual, whereas antimicrobial resistance to bacteria happens in the environment. So less of a problem in that sense. Okay. I'm interested in biomedical science. We just got 5 minutes left, guys, so we'll try and whiz through these. I'm interested in biomedical science. However, I need a grade 6 in maths. I've got a grade 5. I think, unfortunately, I guess you just need to do whatever the basic requirement of the course is, but you should just, it's worth talking to the admission office and seeing if there's anything that they can do to help. How much of the course is focused on immunology versus microbiology? I'd say it's more focused on immunology, but certainly microbiology is a feature. And in your last year, you can do much more microbiology if that's what you're interested in, which tends to mean the study mostly of bacteria. But we also support people who want to study fungal infections, virology, obviously. Sorry, Andy, I'm rather whizzing through this. Climate change, how it impacts infections and scientific discoveries. So yeah, I think it'd be a really good course for that. And although it would probably be a small part of what you'd be studying, it would give you the skills to go on to do a master's more focused on One Health or climate health. And certainly is a really good foundation, I think. Absolutely. Takai has asked, how can infection cause chronic disorders? For example, H1N1 cause type 1 narcolepsy. I'm not familiar with that association between flu and narcolepsy, but again, really, it's the virus that tends to cause an immune response that becomes dysregulated or over vigorous, and then that can lead to disease. So one example is multiple sclerosis after Epstein-Barr virus, so EBV infection. And we don't really understand why that's the case, but it seems to be a very strong association between EBV and MS in some people, for example. Um, I'm interested, I guess that's in CRISPR. Do you know any easy to understand resources? I think generally if you just go on YouTube and type in CRISPR tutorial, you'll probably get a decent lesson there, but we'll teach you about it if you come to university. Autoimmune disease, organ specific, but affect the whole body. Yeah, good question. So it depends which part of the immune system is activated, and if it's a bit of the immune system that tends to be in your whole body, then you can end up with really severe disease everywhere. But autoimmune disease is actually super complicated. So one autoimmune disease can behave very differently to another one. But yes, we have lots of people working on that in our division. You learn a lot about that if you came here. Um, Interesting areas of research you recommend to explore in our own time. Come back to that in a second. I'm sorry, I'm rushing through these. I'm trying to make sure we do all the course ones. Could you talk a bit about the project? Yeah, Andy, do you want to talk about your experience of the project as BSc and then maybe MSc? So,
Uh,
yeah, being part of a team, etc.
yes. So essentially, you become a member of the lab of the supervisor. And how that was done was that you get sent a form where you fill out some of your interests and you discuss that with the course lead, and then they will we will come up with an agreement essentially to a supervisor, and then you would talk to the supervisor to agree on a topic that you would work on, and then that would be your project for the year. Um, so what was it? Do you become, yes, and you can continue on with the same research. So that was exactly what I am doing with my master's currently. So I was continuing on my undergraduate project.
Yeah.
Uh,
And the idea is you really get stuck in and you come to the lab meetings, you do your own work,
yes.
but at the same time, you're obviously getting lots of supervision and support. But the idea is, as Andy's done a great job of, you actually do a lot of your own experiments, generate your own data, your own questions. Yeah. And there's a lot of questions about assignments. Yes, there are written questions. I mean, Andy, do you want to very quickly whiz through year one, year two, year three in
I
terms of the
think
balance
we're
of
actually running out of time and might have to leave it there.
Yeah, I mean, bottom line is yes, there are written one. There's written exams, but there's also some coursework, and there's also um, modules that are examined through you giving a presentation. So a lot of different things. We give you a lot of training and support how to do that. Um, any books? Again, I think Andy's given you a couple of good tips of websites. But nothing is. If you're interested in it, go for it. That's the key thing. What makes UCL different from other universities? I mean, I'd say there isn't really that similar undergraduate specialty course in infection immunology. Give you the breadth, but there's also that real specialist discipline. I think we're going to disappear in a second
Yeah,
because this just suddenly signs off
we'll have to
abruptly.
leave
But
it there,
I'm sure you can get hold
unfortunately.
of us through the admission office if there are other burning questions. But yeah, really some really thoughtful, interesting questions there. So that was great. But yeah, you can come through Unibuddy, I think, as Kate said in there in the chat, and if there's something you really want to ask us. But yeah, really thoughtful questions. So hopefully see a lot of you soon.
There will be a short survey after the event closes, so if you have the time to fill that out, it would be amazing. Thanks everyone for your comments and questions, and thank you, Dr. Fink, for an excellent session. Have a great evening, everyone.
Cheers, Andy.
Cheers.
See you on, well, Thursday, I expect, in our
Thursday.
lab meeting.
Yeah, yeah.
Bye, everybody.
Bye.